All right, so we got atmospheric nitrogen. And so the concept is that the biosphere requires nitrogen. We have 80% nitrogen in the atmosphere. We can't get it, right? I can't convert atmospheric, can't nitrogen in the gas form. I can't assimilate it, right? Even though I need it, I can't do it. So plants are responsible for this conversion of nitrogen to ammonia. And then once it's in ammonia, we'll talk about how that changes. So we have this process. This is a process that is catalyzed by nitrogenase. So last time, we had this nitrogenase enzyme. And it needs to be reduced. And we talked about how this is the active form, nitrogen. If we have oxygen around, then we get nitrogenase. It's bound to oxygen, and this is inactive. Active. And so in order for us to get back to this active state, we have to add leg hemoglobin. That's what we talked about last time. So leg hemoglobin oxidized. Now it'll take this. Leg hemoglobin is reduced. And then this reduced leg hemoglobin will donate this to the electron transport chain. So and so the we shouldn't put reduced or oxidized here because it's very intermediate. Take that out. You probably already have this in your notes. And then the electron transport chain takes this and reduces it to water. And so what we have is a situation where we have an electron transport chain in the bacteria that requires oxygen as being the ultimate electron acceptor. This makes sense because it's trying to keep up with the metabolism of the plant. Plant is going to be providing a lot of sugars. What the plant needs is a lot of this nitrogen. If the bacteria were fermenting or anaerobically respiring, and they weren't using oxygen as a terminal electron acceptor, they would be growing at a really reduced rate. And so the ammonia being produced wouldn't be able to keep up with the growing plant. Effectively, the bacteria would be the limiting thing. And they are already, because nitrogen is the limiting thing. So this is a kind of a creative way for the plant to create an enzyme that is able to escort oxygen as it enters the cell and deliver its electron transport chain without any other biomolecules being indiscriminately oxidized along the way. This protects nitrogenase. We have a suite of enzymes in our cells, as do a lot of bacteria that are, dealing at the aerobic interface that take care of this oxygen. We have electron transport chains. If you put oxygen straight on those electron transport chains, they're going to the electron transport chain is going to be oxidized by oxygen if it's not participating as a terminal electron acceptor. We have a lot of metal-containing enzymes that are catalyzing reactions that we need for a lot of different purposes, DNA binding proteins, whatever. If oxygen was just around, just floating around in the cell, we would have those enzymes continuously be in an oxidized state and inactive. And so oxygen is really good as far as it being able to offer a high energy yield if it's used as a terminal electron acceptor. In pretty much any other case, it's very toxic. So this is the first advent with this symbiosis that we have evolved an enzyme that honors two things. Well, oxygen is a problem, bind to oxygen, and use oxygen so that we can make more energy. Okay, that's just a little review from last time.
Dr. Cruz, real quick. So this is aerobic bacteria because it's used oxygens.
Yes, very important point. Yes.
Dr. Cruz? Could I see the corner of the board over near the leg hemoglobin? Like I can't see the last arrow. Like on the far right. Is there anything over there?
I think that's the edge of the board.
Okay, yeah, the ET part. I can't see that.
I think someone mentioned last time that this could be a function of, is that better, guys?
Kind of. I still can't see the far right.
So somebody said before that it's the function of the way you're using your screen.
Okay, let me try and switch it.
Like if you put it on.
Oh yeah, okay, that was it. Okay, thank you. All right, never mind.
Hold on 1 second. I have a cup of coffee calling my name. Okay. All right. So now turn to my beautiful face. So we have, we'll go back to the board here. So we have the symbiosis where the plant requires the nitrogen. So if you can imagine like all these roots, you know, we have nice plant trying to grow, trying to eat 3 leaves. Okay, so we have a nice plant that's trying to grow. Hold on, let me shut off that light. Is that better or worse?
Much better.
Worse, guys. Better. That's better. Weird. For me, it looks worse, but I don't know. So we have the roots here, and the roots here are going to require some nitrogen. So we have bacteria over here nearby. Got to get these guys together. And so there's going to be chemical signals that are going to be required. So the plant is going to put signals into the soil, and then the bacteria are going to proliferate in that direction. And then once the bacteria are close enough, the plant will sense that the bacteria is going to be emitting signals too. And once the bacteria are close enough to this root hairs, then both species will then start to change their morphology to help establish a nodule. Okay, so this can be probably best. You guys can see my PowerPoint? No, you can't. Let me share my screen. This is what the root hair looks like after inoculation. We have this this bumpy pattern. If you go outside and you pick up some clover, gently get the roots out of the soil, you'll see this. That's nitrogen rich. Keeping all the soil rich for your weeds. So and I'm going to write notes on this, but the the species on the bacteria end are called rhizobia. And this is a specific thing. So different species of clover, alfalfa, whatever legume type plant, they're going to have their own species. And so this is species symbiosis. And when we talk about pairing these two species, we call it a cross inoculation group. Okay. So I don't want to go through all of this as far as writing it on the board for obvious reasons. It's a little bit unnecessary. It's here in the slide. So in your notes, you need to make sure that you highlight figure 2310. This is a really important figure. This is going to definitely be on the exam. And it's easy points too, because it's just steps, right? So I'm going to explain the steps to you, but make sure that you put this in your notes is really important. This is how bacteria and plant establish a nodule formation, okay? And so it begins with, let me see if this is true, step recognition. Okay, so as I said, as I discussed before, this is the beginning, but this is after there's been secretion from the plants, and I'll write this stuff down. Plants secrete organic molecules that stimulate the cross-inoculation group. So there has to be bacteria knowing that the plant root is there. And then once the plant has secreted these organic compounds, and then the rhizobia sense these things, and now they're in close proximity, then we can establish these processes. Okay, so I just want you to, in your notes, make sure that you put that before this there is, you know, chemical signals being received by the plant and the bacteria. And now we have the bacteria touching the root hair, and we're going to start this process after. And I'll write these things on the board, the before step one here. So we have recognition and attachment. So in purple here, you see a rhizobial cell. Okay, this is the tip of a root hair, and so there's going to be receptors on the root hair, and these are going to bind to the bacteria. This is something like, you know, we have the like COVID and the epithelial cell, how there's a specific cell-to-cell contact based on receptors. This is the same kind of thing. This is why this is species specific. Bacterium secretes nod factors that cause root hair curling. So this, so the thing that you need to remember is that what we're looking at when we're looking at these cells on slide 2 is that we're looking at the plant cells. And so the plant cells are changing morphology, and that they're changing morphology because they're sensing factors that are coming from the bacteria. They know the bacteria are close. They know they're in high concentration. Why? Because the bacteria are secreting nod factors. And then when the cells, the root hair cells, are engaging with these nod factors, it stimulates a morphological change that's called root hair curling, okay? So you get this kind of curling pattern, and the bacteria continue to grow. And in this loop, in the root hair, that's curling, you can now get an infection that is encompassing. So in step 3, you can see how we have an infection thread, and now in purple, you see this is like kind of how bacteria. This is the bacteria now that they have in purple, and the bacteria actually start to penetrate into the root hair, right? The roots through the root hair, I should say. And so then the bacteria are growing into the root cell, and then what you have in step number 5 here, these nodules, that's the product of these interactions. The bacteria take on what's called a bacterioid state. And this bacterioid state is kind of a irreversible kind of zombie state where it stays locked in a metabolism, and they don't, if you try to coax cells out of a nodule and try to plate them, they don't go. They're stuck in this state. They're irreversibly and engaged. They are irreversibly engaged in a symbiosis, and so Know these 5 steps, very important.
Dr. Cruz,
Yes.
can I ask a question? So why does plant needs nitrogen?
Oh, because, and this is kind of a simple question, you know, and it seems like, what's the big deal? Just simply because, here, I'm gonna, you guys can see me? Because we have, the plant is doing this, right? That's what the plant's doing. So, and then from this glucose, we can make DNA, we can make proteins, we can make fats. Problem is, is that there's nowhere in here in this H2O to CO2 to glucose that has, I mean, every protein, I mean, we talked about proteins. There it is right there. There's a nitrogen. If I draw DNA, you'll see even more nitrogen. So these are nitrogen, so every time you make an amino acid, every time you make a DNA base, you have nitrogen. So this nitrogen has to come in from somewhere. So we need it for the proteins, we need the DNA. Some fats have some nitrogen in their the attachment group at the end. But proteins and DNA are nitrogen rich, and so we need nitrogen continuously as we do this. So we need a way for the plant to biosynthesize. That's why. We do too. We need nitrogen just as much as a plant does. We get our nitrogen primarily through glutamine in our diet. And we can get it through arginine, but mostly it's glutamine. That's how we assimilate it. We have pathways that will convert glutamine straight into DNA and proteins. So that's why. It's just a really big fundamental. The biosphere needs nitrogen too.
Thank you. Got it.
So this is a really important thing to know, because what you're thinking about is, oh, how does the biosphere get nitrogen? Like every bacteria cell, every animal, every plant, everything in the biosphere has these nitrogen. Where does it come in from? That's how it comes in. It comes in through these nodules. There's some other ways, but for the most part, it comes in through these nodules. I think I said 30%. I don't really want to go back in my notes, but anyway. So now I'm going to just write some of the notes that, because the book goes on for quite a bit about some of this. So I'm going to just capture, you know, Dr. Cruz's favorite hits here. So each leguminous plant has a specific cross rhizobial species, also known as AKA cross inoculation group, as we say on the street SIGs. Okay. Plants secrete organic compounds that stimulate SIG growth. So these bacteria, these rhizobial species, are going to have a tendency to want to go near plant root hair cells because the plants are providing nutrients that make it easier for them to grow. And so they're going to go towards the plant. It's what the plant wants. We have all those steps, so I'm not going to go through all those. Plant cells divide and surround rhizobia to form nodules. Okay.
Just out of curiosity, are the plant cells dividing and going around the rhizobia as a form of their own stimulation, or is that the rhizobia stimulating the nodules? I don't know if that made sense.
Let me like you're kind of, let's put this next thing up here.
Okay.
And maybe it'll make more sense.
What does that last line says?
Plant cells divide and surround rhizobia to form nodules.
Got it. Thanks.
Yeah. So we have nod genes. These are rhizobial genes. That produce proteins that direct nodulation. And so this induces hair curling and triggers plant cell division. So that might answer your question a little bit. So
Yes.
the plant emits organic compounds that are nutritious for rhizobial species. The rhizobial species will start to grow, divide, prosper, and then these rhizobial species will produce the products of nod genes, these proteins. And once the plant has when the plant is encountered with a high concentration of the products of these nod genes, it's going to then change. These things act like a hormone inside the plant cell, and they induce hair. It's like a hormone in a way, induces hair curling and triggers plant cell division. So this bottom part, plant cells divide and surround rhizobia to form nodules, is what plants do in response to nod genes.
It seems like the roots would want to grow towards nutrition of some sort, it's like a good evolutionary, concept to have.
The roots are because plants don't really have a lot of, they don't need organic matter at all because they're organic. Their carbon's coming from CO2. If you were to weigh out how much they consume as far as from the soil, it's not really that much. They're just trying to get minerals, and they're trying to get nitrogen. They're trying to get all the things they can't get from the air and water. They need water, but it's modest. Think about a cactus, right? I mean, how much can come into a root anyway? I mean, it's not much. So they need this, yes. And so the plant root is going to be absolutely going towards all nutrition. Now, let me read my notes for a second.
Dr. Cruz plant doesn't have immune system like us to reject the bacteria. Like some
They do. Yeah, they do.
They do?
Oh yeah. Yep. Plants are like always freaking out, man. They really are. I mean, to get a plant to grow productively is actually difficult. They're constant. They have so many stress response genes because they're very upset if anything comes in contact and invades them. They get infected. It really it hurts them. They get viruses just like we do. Um, Microbes will try to kind of tap one of their veins and get up into the leaves. They have a lot of problems. So they do have an immune system. It's not like us where they have generation of IgGs, I don't think, but they do have a response to foreign antigens and they can clear them. It's kind of amazing that plants can stay healthy, considering that they're just putting themselves out there. I guess we put ourselves out there too, though. All right, let me see what I want to find out. Well, okay. So now we'll say root nodule events. The book doesn't call it this way. So we have nitrogen fixing bacteria into fixing bacteria that require the plant to supply an electron donor that come over here require the plant supply an electron donor, which is just another way of saying energy source like glucose or whatever. So, which are organic acids So these are being given off by the plant. So this is going to go in the soil. It's going to promote bacterial growth. And so we're going to, in addition to any non-plant based fermentation that's going on in your garden bed, we have the normal plants trying to deal with their own bacteria and organic acids. That's why you have to really be careful to not promote bacterial growth in your soil because the pH will plummet, and then you're going to have to treat with lime to raise that pH in your garden bed. Okay, this is why this is a constant issue with the pH. Succinate, malate.
Dr. Cruz,
Oh yeah.
when it says which AS organic acids, I'm sorry, I don't know what.
It's just an acid that's organic. So I'm not trying to be facetious. And so you've seen this before. You just probably didn't realize it. So here we have an organic acid that's pyruvate, right? We've seen acetyl-CoA just has a CoA to it. So here is, that's acetate, right? So acetate basically goes into the TCA cycle. Pyruvate is produced after glycolysis. So these are organic based, these are carbons. Very good question too. And then they happen to have a carboxylic acid groups, and so they're called organic acids. So the thing is to know that, oh, okay, a common product of a lot of the bioprocesses, energy yielding bioprocesses, are organic acids. You probably never thought of them that way. I didn't say anything like that, but that's what's going on. So succinate, malate, fumarate are all organic acids. So then the process, So that's what the plant wants to offer: succinate, malate, and fumarate. So then how do we process these? Organic acids are one, oxidized by the TCA cycle to produce energy. We talked about that. Produce energy. Or two, converted to pyruvate, which is then oxidized to reduce, not produce, reduce nitrogenase. Nitrogenase, we talk about how oxygen inactivates nitrogenase. Nitrogenase is inactivated by any oxidation. And so the minute, and nitrogenase converts into the atmospheric nitrogen to ammonia. And so anytime you have nitrogenase function, it becomes oxidized. And so it continuously needs to be reduced in order to function. And so these organic acids are a source of energy, but they're also a source of electrons for maintaining nitrogenase to be reduced so that it can continuously convert N2 to NH3. So another way to put this is, so if we were to put number one, the example of how that is, we can have malate, succinate, and we've seen this show before, right? Fumarate, and they are going to go into the TCA cycle, and then that is going to yield energy. You can make a living off that. I don't know if you guys can see this. Let's see. Let me try to raise the
I was
board.
gonna say if there's any way to pan the screen down a little bit, that may help.
That's better?
Yeah, that's great.
Okay, cool. So that's number one, right? So that takes care of the microbes' energy needs. But it needs to give back too, right? And so in this other scheme, we have pyruvate. We have pyruvate that is converted to acetyl-CoA. And then we have nitrogenase, which is oxidized. And then we have nitrogenase, which is reduced. I'm going to rewrite this part. We have organic acids. They become pyruvate, right? And then pyruvate becomes acetyl-CoA. we know that this is an oxidation. We are normally seeing this with pyruvate dehydrogenase, and we know that in this process we make an NADH. So we know that this is an oxidizing event. In this case, though, what happens is this pyruvate is used to take nitrogenase, which is inactive, and we can put a little oxidation in here, and then we can say nitrogenase. It's reduced, and now it's active. And in its active state, nitrogenase is going to take nitrogen and make ammonia. And so in this process, you guys can see this okay? Yeah, okay. So pyruvate is oxidized to acetyl-CoA. These electrons go on to reduce nitrogenase. Nitrogenase needs electrons to convert nitrogen to ammonia. So we're continuing. So the plant is supplying these organic acids. The organic acids can be taken into the TCA cycle, and then the bacteria can make a living. But then some of these organic acids have to serve to be electron sources to convert ammonia, nitrogen to ammonia. Okay. Any questions there?
Yes. Yeah. So Dr. Cruz, when nitrogenase with oxygens is inactive, so I thought that means nitrogenase is reduced by oxygen when it's therefore is inactive.
This is just oxidated. This means oxidized, not oxygen.
That means it's lose electron.
That's right, lost electron.
Lost electron, and if it's gain electron, then it's active.
That's right.
And so oxygen is just acting as an inhibitor. Put the enzyme into the RBT state.
Yeah, I mean, so what would happen if we had oxygen here? It would just be here.
In my screen.
So oxygen, if oxygen were in this player, then it would take those electrons away from nitrogenase, produce hydrogen peroxide. That's totally, that's not physiologically relevant. This is toxic. It'll start oxidizing all kinds of molecules. We don't want that. We don't want that. But this is how oxygen interacts with nitrogenase, if it's allowed.
Oh, is allowed, but
like hemoglobin's binding the oxygen, so oxygen doesn't have free access to the nitrogenase.
Okay.
Mm-hmm. If we didn't have like hemoglobin, then that would happen. And the oxygen, then this would happen. But like hemoglobin's binding to the oxygen, so the oxygen's not doing that. And so instead, then what we can do is we can donate these electrons to nitrogenase, and nitrogenase then can convert atmospheric nitrogen to ammonia.
So the whole thing is in the plant, right? The
this is inside of the bacteria cell.
Okay. Yeah, it's nothing.
Bacteria cell. So here, let's review. So in the beginning, we had a plant cell. And now we can call this a rhizobial species now that we know more. Right? And so we have CO2 making organic acids. Using sunlight here. This is a little updated now that you guys know more things. And then these organic acids go to the rhizobia. Rhizobia we can say that the N2 enters rhizobia, is converted to NH3. NH3 leaves, goes into the plant. And then what's going to happen is inside of here, this is what is going to go on next. Glutamate. This is, I don't have a lot of space to work with, but basically what's going to happen is glutamate plus NH3 equals glutamine. And now this is the form that most life forms can take in. So the plant offers photosynthesis, production of organic molecules, rhizobia take these organic acids, make a living, and then with some of this organic acids has the ability then, because it's making a living, to take in nitrogen, convert it to ammonia, and the ammonia can go into the plant, and then the plant can convert the ammonia to glutamine. Is that clear?
Yes, so the bacteria cannot live without hemoglobin because it's become toxic?
Yeah, it needs like hemoglobin.
Okay, cool. Thank you.
Yeah, and then so the plant, I have another concept that was used, takes ammonia and uses glutamine synthetase. I used to work on this enzyme as an undergrad to assimilate ammonia. Assimilate you could say ammonia. There was nothing wrong with that. I just, in my head just now, I'm like, well, the nitrogen, because it's about the nitrogen, not really the ammonia. And so what we have is we have glutamate, and you don't have to write the structure. I will. I think it's interesting. I'm like running out of space. I don't know if you guys can see this. And so then glutamate plus ammonia goes to,
Dr. Cruz,
yes.
I'm sorry to interrupt you, but my screen is coming and going. Is everybody else is working? Can you all see the screen, or it's
Mine
coming
is
and
not
going for you
working.
all too?
Mine comes
Mine
in
looks
and out as
fine.
well. No, mine is in and out for some reason as well.
Yeah, so it's not
Mine's
just
been
me
going in and
then.
out as well.
All right,
All
let's see.
right, it's not me then. I'm on mute.
Give me a second.
I can't hear you.
Well, I just tested my internet speed. It's like incredibly fast.
Yeah, no, yours is good. My internet is kind of fat and not my screen was not being cut.
Oh, so it's on that end. Oh, so you've been okay.
Yeah.
Okay. Yeah, I have fiber optic Google, but I want to make sure everyone's okay. So can you see this right now?
Yes. perfect.
But anyone else, I want to make sure.
Yeah, that looks good.
Okay, I'll try to not go too fast then. The erasing might be stressful. So glutamate, this, and so then when you look at, I'm going to go to the top. I'm just going to rewrite this. We have glutamate, glutamate structure. And this is not, I'm not going to test you on this. I just know there's some molecular dorks out there that like this kind of stuff. So I'm just putting it here so that you can see it. If you're not a molecular dork, you should be, but it's okay if you're not. You don't have to self-shame if you're not into the organic molecule drawing. I think it's just more, when you're looking at this stuff, it can be a little bit disorienting and you're not sure what you're supposed to see. And then you can get down on yourself. You're like, man, all these atoms and molecules, just too much. I don't understand what's going on. And then you go on through, a little existential crisis. But really, when you're looking at organic molecules, just let go. Try not to think of it too much, and see if you can observe something that has changed, and just notice this as a thing to observe that can happen, rather than thinking that you're supposed to be able to reproduce these molecules like I do. Like, I've been doing this a long time. I mean, it's just, I don't think I was always able to do this. So I'm just showing you this because I'm trying to show you an event that's occurring. And I bet you that if you saw, when you look at a reaction, you can just do it like that, where it's just an event that you're observing. See, and so what the simple issue was, we wanted to be able to take all the nitrogen that's in the atmosphere, we wanted to bring it in the biosphere. What that's going to mean is that we're going to want to take nitrogen and put it on some molecule so that now we can process that molecule no matter what organism you are in the biosphere. And so what we have is we have NH3. NH3 was N2, right? So we had a bacteria that converted nitrogen into ammonia. In the plant now, and this is inside the plant, now the plant has glutamine synthetase. And it can take this glutamate. You can see there's some nitrogen here, right? But it's found a way to take this ammonia, and now it appears here. That's the interesting part. You don't have to memorize biochemical pathways, but you can say, oh, we've made a molecule that has the nitrogen there. This, 99% of the biosphere can deal with that and incorporate it and turn that into either amino acids, or it can turn it into DNA nucleotides. Okay. And this is glutamine synthetase. I can't spell, of course. So there's a substrate missing here. There's enough information on this board for you to be able to tell me what that is. Can anyone tell me what's missing? Glutamine synthetase.
ATP?
ATP,
ATP.
How'd you guys know that?
because it's a kinase and they require ATP.
You have synthases and you have synthetases. Synthetase means requires ATP hydrolysis. That's how you know. So it's a synthetase, meaning that its synthesis requires ATP hydrolysis. Kinases phosphorylate things. Nothing's being phosphorylated here. Not really. I don't want to get into it. I can draw the whole mechanism. It's Not necessary. Let's get away, step away from the board, sir. Okay. I'm going to share my screen again. So we have the cross inoculation group. Just make sure I didn't miss anything. Pretty pictures for you guys. Okay, so I'm going to briefly cover this. We have other symbiosis. I'm not going to go through all the symbiosis in the chapter. It's not, I think once you have the theme, then you can understand what the concepts are with symbiosis and how there's, you know, more of a relationship about offering nutrients to each other or some other thing like hydration or whatever. So the mycorrhizae, what's that? On the shiznit. All right, stop it, Dr. Cruz. All right, so mycorrhizae, Snoop Dogg symbiosis. No, I'm just kidding. Mutualistic association of it's plants and fungi. It's not just bacteria. So this is a fungus. Fungus are eukaryotic, right? And so the remarkable thing about fungus is that they really have good enzymes for just tapping into the nutrients. Remember last time I talked about how we have you know, logs that are sitting out there, and that how is a log sitting out there, but if a body, you know, if an animal's there, it's gone within a week, and a log can stay there for years. And the difference is because the structure of a plant is such that it's really difficult for a microbe to find an enzyme to break that down. Well, one organism that does have that is fungus. So fungus, and said, well, why would a plant want to deal with something that can kill it? Well, it's not that. It's just that the fungus has access to a lot of the biostructures in decaying matter and can break down things and release those nutrients. And so there's a mutualism that's potentially useful for both parties here, because the fungus still need glucose. And so if you can find a way to be mutualistic with a plant, and the plant can, so the fungus can break down minerals and offer it to the plant can then offer sugars, and then the fungus can grow. And so that's the basis of this mutualism. And so this one's really simple. Okay. If you have how we spell this wrong? His A is easy. All right. So what you have basically is you have a fungus, plant, okay, and fungus offers lots of things. In the book they talk about phosphorus, but it really depends on what you're talking about. And then the sugars, give me some sugar, the plant. Okay. So that's a mutualism. Very simple. Okay, I'm going to go back to sharing screen. So we have coral reefs too. Coral reefs are, they have a symbiosis with, you know, bacteria as well. They include cyanobacteria, chlorophytes. Dinoflagellates is the big one. Diatoms, a lot of stuff. So here's a profile of that, of those organisms. And basically what happens is, and I don't want to get too much into all this, but when you have this kind of symbiosis, you have an opportunity for these bacteria to be immobilized and gather light, right? If they were just floating out in the open ocean, that's much different than if you're, you know, immobilized on a definite structure. And so then the organisms, the cyanobacteria, can be within the coral, do photosynthesis, and offer sugars to the coral. Really good deal for the coral. And so both parties benefit. And the corals reproduce sexually. It's a little reckless, right? They just put it out there in the ocean, releasing gametes in the seawater. But obviously, the higher density of corals that you have, the less random that is. And so they really need to have quite a bit of a structure. That's why you see them organized in reefs. The corals regulate the cell division of these bacteria, and so that symbiosis is controlled on the eukaryotic end of the coral. And some of these bacteria can be ingested. Corals do have primitive guts. Coral bleaching occurs when you have snapped this symbiosis, and it usually comes from the bacteria being unable to tolerate the conditions, whether it be too intense sunlight or too much heat. Usually it's about the heat, but the UV index and a lot of these things play into it. And so when you have global warming and responsible, you have to remember global warming is about energy and heat, not temperature. And so there's a lot of intense energy that's going on, and that's making the conditions a lot more as far as UV light as well. So this has an effect on the bacteria, and when the corals lose that symbiosis, they're not getting the sugars they usually get from this photosynthesis of the bacteria, and then they die out. It's really too bad too. I mean, let's see, I'm a certified diver. I've dove into the Keys. Actually, diving is how I discovered my love for biology. Ultimately, it was illuminated from that. I've always been, if I was a kid walking around wondering why logs weren't breaking down the same thing as another animal, then obviously I had a biology in me the whole time. But going into the marine habitat was very stimulating for me. I just loved it. And there is a big difference between the way it was in the mid 90s and now. It's remarkable. So if you have a chance to go in there, and you don't have to be a scuba diver, I would recommend if you just went down to the Keys and you just go snorkeling. If you find a really good reef, you'll have just as much fun as if you were scuba diving. So
Wait, Dr. Cruz, can you add one the coral slide?
what about it?
Yeah, I think we're missing
What
that
is
slide.
lysis of symbiont? Yeah, no, not this one, the previous one. No, like there is a loss of color in coral because of lysis of symbiont.
Symbiont.
Yeah, what is that?
Symbiosis. It just means that the symbiosis, you can call each one a symbiont, and so the coral isn't bleaching because it's dying. It's bleaching because it's losing its bacterial partner, and without that, then the coral dies. So it lyses the symbiosis. Maybe that's the best
Got
way to
it.
put it.
Yeah, thank you.
Yeah.
Dr. Cruz, I think we're missing one of the slides, the one with the coral bleaching.
In your PowerPoint supply,
Yeah, it's missing.
I will, I just worked on this today, and so I'll upload the new this chapter 23. I'll replace it.
Okay, thanks.
Yeah, I just worked on this today, and I normally don't have the coral bleaching. I added it this morning. Um, I was working up early this morning. I'm like, well, I bet you that would be nice to know. You don't have to know all these things about all the coral bleaching. Okay. All right, let's go back into.
Dr. Cruz, I think you're muted.
All right. Now we'll talk about. So these are all symbiosis. This is all friendly stuff. There's um, no parasitic relationships going on yet. And I know that in the book, it talks about dental care. I know some of you out there, waiting for me to get to that. I'm not going to go over the healthy flora of the oral cavity. I'm just gonna, when we go into the parasitic stuff, then it's just easier, just presentation wise and the way the information flows. It's just easier for me to talk about tooth microflora in terms of a pathogenic thing. So we're going to talk about mammals now. mammals as microbial habitats. Microbial habitats. This is not trivial. I mean, this is, your microflora in your gut dictate a lot of your metabolism, your stability, neurologically, not just your mental, but also how stable your gut is and how food passes through you. So this is a symbiosis that's been cultivated and selected for over millennia. And so when we have the flora of your intestines, there's, I'm gonna have to go on a little, illuminate my face because I'm about to talk for a little bit. And so when you have, I've talked about before how you have the flora that are inside of the primitive gut of all these very small multicellular organisms in marine life. And then as we have gone on, then you start to develop fish, then you have reptiles, and then you have birds. You go through evolution, and through all of this, bacteria, a part of health and viability. If you disrupt this, it can really injure the organism. So one major difference in microbial flora in your intestines, it occurs between carnivores and herbivores. If you're a carnivore, then you're going to have an intestinal structure that's very simple because it's really difficult to break down plant matter, as I said before. So if you're an herbivore, you really are in a condition where you have to spend a lot of time digesting all of these things, and you're going to have a very complicated microbial profile. And so this is the reason why when you look at herbivores, like if you look at gorillas, you look at cows, sheep, if you look at their guts, monkeys, they all have very big guts. And the reason why is that they have a lot more intestines and they need more surface area in order to extract nutrients from these plant-based meals. And carnivores, since they're eating meat, they can digest what they have to pretty readily. Longer time in the intestines isn't going to make a big difference on whether or not they can digest something. Plus, having a big gut isn't really good if you're a carnivore because you need to be fast. And so they have more streamlined intestines. And so herbivores really have much more complicated intestines. For us, it's a little different because we cook our food. So we don't need all that time. We can just, if you cook plant-based food, it breaks down all those cellular structures that would normally need to be assessed enzymatically. And so we don't really fit in so neatly into that. We're kind of, obviously we're omnivores, but even if we're all plant-based and you're like, well, we don't have these large intestines, we're not meant to be plant-based. We cook our food. That's the difference. Okay. All right. Enough of that tangent. And so we have mammalian, have a mammalian gut. So the reason why I went into all that is that we see differences in the structures of mammals along these lines. And every mammal needs to be able to cultivate microbial growth and fermentation. But the extent from which they do will differ based on their diet, okay? So mammals have intestinal structures structures that cultivate oh yeah, we are almost, it's almost time to go. Cultivate fermentation. Of plant material. Okay. Herbivores depend. I have to do better on my E's. They really slipped over the past decade. I still can't do it. I don't know why. Depend on fermenters for energy requirement. And then, I think you guys can see this. Let me see. So plant material or I should say indigestible, require one, anoxic fermentation chamber. Sounds heavy duty. I'm going to start this up the top again. These small boards, okay. So I'm going to put herbivores have anoxic fermentation chambers. Okay. And then two, and this is for plant material. Two, extended digestive time. So now I'm going to go over, and I know our time is almost up. It should be up, but hey, I know that you don't have to go anywhere, so I'm going to keep you for a couple minutes longer. I just want to share. Hello. Okay, you guys see me okay? Okay. So we have herbivores. I think you guys already know these definitions, so I didn't really want to write them down. I have herbivores, carnivores, omnivores, and you guys can read most of this, but basically you have structures that are going to be required in order to have okay, here's the intestines on the left. You're going to have regions in your digestive tract that allow for these bacteria to spend time with the food and break it down. They can't just start going through the digestive tract. So on the left, you see what we have is a foregut fermentation chamber. And then we have this cecum, which is a hindgut fermentation chamber. Both of these are designed to be little places where bacteria can really spend time with the food and allows for some of this material in the intestines to come out of not circulation, but at least out of movement. And so all of these animals on the right have these kind of structures. And they're called ruminants if they have the foregut situation. And so here, I don't know if you guys can see my cursor that well. Laser pointer. Okay, so you guys see my laser pointer. So this is how this goes, and this will be how we stop this session. And I will have a quiz on Thursday. I totally forgot about the quizzes. I'm sorry. I'm just trying to get in rhythm here. So this is the cow. Okay, so what we have is we have food goes in through the esophagus, right? That's part of your throat. And then what happens is it goes in to this this structure. And then you will actually have this food kind of spend time in here, and it actually can come back out. If you see, you can see cows chewing and they haven't eaten anything. And what they're doing is that some of the food that went in to the esophagus spent time in the rumen, and then it comes out as cud, and then it comes back in, and then it starts to go through the small intestines. And so food that goes into the rumen will not enter the small intestines until it's broken down to a certain time. And so this will come back out, and then the cow will rechew it, and then it'll go back in, and then eventually this will be small enough so where it can go into the small intestines. This takes a while. And here there's cows with these plastic devices. You can reach in and grab a culture and study them. That's awesome. And so we can go through more of this next time, but effectively what happens is we start with food on the top, then we break down into the smaller components, starch and sugars. And then once we have these sugars, and it takes a while, right? You have to chew up the cud, you get it through there, but once it's through the small intestines, what happens is these small sugars then are absorbed into the ruminant bloodstream. Okay, so I'm gonna all right, you guys should be able to see me. Giant cow petri dishes, effectively, yes. Um, so, yeah, I kind of worked on some of these kind of guys last time. And so all I really wanted to talk about was those two slides. I'll recover that last time. All of the quiz material for Thursday, and I'll put it up very soon. It'll contain maybe 2 quizzes. I'm going to go over the symbiosis, basically chapter 23. Basically chapter 23. That's what I'm going to cover. All the stuff so the microbial mass is not part of chapter 23. So I'll just stick with chapter 23. I probably will make it 2 quizzes. I'll probably have one on the symbiosis, and then I'll have one on just plants fixing bacteria, the root nodules. Okay. And I'll have that out tomorrow morning, so be ready for that. And if there's any questions, let me know. What's going on? I know some people have a class at 12, so I'm going to go ahead and end the session. When this is released, I'll have it available for you, hopefully in the next 24 hours. I'm sorry if that's not the case. Okay.
Dr. Cruz,
Yes.
so I did send you some slides. I don't know if you had a chance to go over them with some microbes, malaria microbes.
I can try to look. So this is you emailed these to me?
Yes. Yeah. Thursday.
I can look for them real quick.
Okay.
This is for your stuff. Okay. So why don't we we'll talk. Just stay on the line.
Okay.
Is there anybody else with questions?
I had one question about the test. I don't really want to start a World War III over it. There was just like one question that I think you actually went over, and it was just asking about the phenotype. And I think the answer that was on the iCollege test was, it was like written in the genotype. I believe the answer like should have been like a lac mutant, but instead it was a lac oxotroph, but it was written.
You got credit for that. You sure you didn't get credit for that?
Yeah, I didn't get credit back for that one. I got credit back for the other one.
Did you look at your points?
Yes.
Oh, all right. Email me. I might have just missed over yours. I think most people got that corrected.
Okay.
This is just a mistake. I corrected most people's, but maybe yours slipped through the crack. I'm sorry if that's happened.
It's okay. I'll just email you.
Yeah.
Okay. Thank you.
Yeah. Anybody else?
Dr. Cruz, would you just like slide that board a little bit on the left? So got it. Thank you.
All right. Okay, guys. Well, then I'll go ahead and, or you guys go ahead and all log out. I'm going to stay on the line with Unu and discuss some of her stuff. So I'll see you on Thursday.
I was just gonna say, I'm sorry I was bombarding you with emails about articles and whatnot. I got talking with a buddy of mine
Yeah.
from who's an ENT doc in Jersey. So he's become a hospitalist in the past several weeks, and then my buddy who does internal medicine in Jersey. He's like, so I'm getting like all this information. I'm trying to process it myself. I'm excited about it. I was like, well, it's novel, and didn't do enough due diligence on my part. But then what was nice is I was able to kind of see these things, get other information, and send it back to where I got it from, and say, hey, you want to examine this? So
Yeah.
that's nice.
And you're trying to go, yeah. I was, and so I'm always very encouraging of these kind of inquiries. I've never seen anything that establishes coronavirus inside of it. It's certainly not necessary for a virus to go through hemoglobin. That's for sure. Um, and this iron dissociation thing, it's like one of those things that makes sense because it answers so many questions for a doctor, but um, doctors don't know about that kind of thing.
Yeah, that's true.
A biochemist would be able to tell you about that.
Right.
I just haven't seen any evidence that there could be associations, but I don't know if I've seen a clear.
Yeah, one particular guy said that, there's a lot of ways that you can get a lot of pathways to having a significant increase in, heme in the blood through destruction of red blood cells that's alternative to this specific virus, but could be um, a byproduct or a secondary result of maybe there's something the virus is doing. Obviously, if you're going to have epithelial tissue damage,
Or
you're going to have.
your immune system, because um, one of the big battles, so when the microbe is invading, one of the big things you do is try to make it yourself an inhospitable environment. You raise your temperature. That's one way, but then also one of the things that happens is there's a big race to get all the iron out of the blood. We have cytophorins that are big molecular cages that basically suck up all the free iron. Because if you you can lower the concentration of iron in the blood below the threshold of which a microbe can thrive, and then they could die. And it usually is the line of defense that works most effectively. And so then knowing something like that, and I'm not an expert in how iron sequestering goes during during immune responses when you're sick. But knowing that there's a battle on that level, and then hearing people philosophize on how iron goes, it just really makes you say, it is not a simple story at all. And you'd have to comb through a lot of information and a lot of tests to be able to really talk about that productively. You know what I mean?
Yeah, and I think a lot of these docs are kind of in a desperate situation to where they're seeing people not make it, and they're like, what do I do? This is a possibility. And there's a fellow that was Maimonides in Brooklyn, where I used to actually do cases, and he was like, I'm seeing things that aren't kind of jiving with what we're talking about. So I mean, I think they're just trying to pin down what could possibly help. I think.
Yeah, the problem is with any medication, it's just so easy to stand behind something when it seems like a good idea.
Yeah, right.
But they can go wrong, and they do all the time. All these
Yeah.
regulations, it's like you see a stop sign, you see a traffic light in the middle of a block one day. What happened? Well, there was an accident. That's why they're putting that up there. The reason why they have all these regulations is there's been accidents.
Yeah.
So, you have to, if you're going to be tested, if you're going to take a drug, usually you have to be tested to make sure that you're compatible. And then once you're cleared, then you can go forward. It's not that the drug can or cannot be effective. It's just that it's inappropriate to administer it in such fast timescales and large quantities. You're going to hurt people.
Yeah, I agree totally on that. Like I said, I mean, when I used to sell pharmaceuticals as well, I definitely sold off label, which is not that good, but it definitely helped a lot of people in certain situations. But this, again, I think what happens is in this sort of, I don't want to say hysteria, but in the absence of scientific, you know, evidence, you do have a lot of superstition, and people say, well, I gave it to this guy, they recovered, I'm going to continue to do that, and that happens all the time. They're still doing it right now out of desperation.
Yeah, so that's the thing in that people, some of these models, and then the models have changed and all this stuff. So they're like, well, they're models. So models are the best we can do. Like this whole thing is like that. You can't have anything. It's ridiculous. Like, well, we don't. If we go out there and don't do anything, we know what happens. We get 1918 flu pandemic. That's what we get. Science brought us to the point where we can at least do some of these measures. If we tested more, we can do ring vaccination and eradicate this like we did smallpox in the 70s. But scientists, see the difference between scientists and normal people. Scientists are very comfortable updating their information. They don't mind being wrong. They like being closer to the truth. People want absolute answers, and it's understandable. They actually want your doctor to be God, is what they want.
Yeah, it just doesn't work like that.
Doesn't work like that. But just because you can't have perfection or definite answers, you can still celebrate that which is closest to helping you. And the closest to helping you is scientific knowledge. Science is going to be what develops a vaccine. Science is going to be what tests all these people. Otherwise, there would be nothing. So, and it makes people feel uncomfortable to not be 100% certain of something. It's weird.
Yeah, there's definitely a fear there
Yeah.
that people don't like.
Yeah. No, and
Yeah.
it's okay. I'm comfortable not being right all the time.
Yeah, that took me a long time to get there because of my previous career. Like, you have to be confident, when somebody's on the table, and even if you're like mostly right, you just kind of have to be confident to calm down a surgeon if they're freaking out. But I agree, this has been a big change for me going back to academia, where if you're wrong or you mess up, you have to say, and it was a hard practice. I screwed up, I was wrong, here's why. But that's a huge exercise in academia after you've been out in like a sales realm or a medical realm where people don't want I think. They want I know, and you better be right about it. So I'm glad I've gone through this exercise. This has been pretty good for me. And by the way, did you get that article that I sent? I can't remember who sent it. It was really awesome. It literally the first few days of your class when you were talking about the beginning of time and the different.
Yes.
That article was awesome, man.
Yeah. I actually, I don't know if I replied to you. I guess I didn't because you're asking me.
That doesn't matter. I mean, you got a ton.
Forwarded that to some family members. They really enjoyed it.
It was
Yeah.
kind of a little bit more, it wasn't as complex, but he's still understandable. It's still Anyway, well, I'll let you get going. I could yammer on forever.
Yeah, I have to talk with Unu a little bit, so let me spend some time with her.
Will do. Okay, guys. See you later.
All right.
Thanks.
Okay, Unu.
Yeah.
I moved Diamond. I don't know what. I moved them to the lobby. I don't know what that meant. I hope they're okay.
Okay.
Seems kind of like what did you just do? You just moved him to the lobby? Okay. So I'm going to share my screen. I have this. Give me a second. I'm trying my best.
So, Dr. Cruz, I just had, kind of like conversation and just listening in. Like, so right now with the whole, talk about anti-malaria medication and how it, the possibility of working with COVID-19 symptoms. So my thought is like we've had like areas whereby people have been exposed to a lot of like malaria medication. Does it in any way help with like building up immunity against the virus? Has there been like anything scientific like that of like in the past that says if you have like a constant exposure to certain medications, what are the risks or the chances of you being infected by certain um, viruses, and does that kind of like help your transition towards the disease stage, or just out of curiosity, really?
Yeah, I don't know about anti-malarial. I don't know the mechanism of, I would have to know the mechanism, and then it's all just database, right? And so so people like, I think I mentioned this last time, like if you're taking a, something for blood pressure, a lot of times it will target the ACE2 inhibitor. And this is the ACE2 receptor, angiotensin receptor on your epithelial cells. Just so happens that COVID, that's the receptor COVID binds to. And so then you're asking, and then you have to wonder, oh, geez, okay, so then does it make someone more or less? I mean, how would that make sense? And the body reacts from so when you take the drug and you have a molecule that's binding the ACE receptors, how does your body respond? Well, it upregulates a lot of the ACE receptors because it senses that there's something going on. This signal's not happening, and it creates a higher density of ACE receptors in various cell membranes. And so then you have to wonder, then you have to think, oh, geez, well, certainly if there's more receptors, if the virus is around, there's more opportunities to bind. And so I think that's true, right? But that's that particular drug. I think in that case, I think you're still better off taking the blood pressure medication. But so it would depend on the mechanism of the drug. That's what I would have to say.
Okay.
So let's take a look at your slide. Let's see. So we have. So first of all, this is about a 10-minute talk. So that usually means around 2 minutes a slide. So that gives you around 5 or 6 slides. You have about twice that. This doesn't count though.
I could read. Yeah, some of them is like the outline and then just diagrams.
Then you can go through this. Okay, so let me see. So why don't you talk me through this as I go through it?
Okay. So basically what my presentation is a topic on the microbe Falciparum, Paramecium,
Plasmodium,
and the causative disease of malaria. So I wanted to kind of zero in on the fact that it's a microbe that causes death with 94% of the cases occurring within the tropical regions. And then actually the strains that cause the actual disease, because there's about 4 different strains of the microbes, but the ones that actually do cause the disease or the highest rate of infection is the falciparum and the vivax strains. Then the introduction just talked about some of the symptoms that are consistent with the malaria disease, how it affects the kidney. It's also a disease that targets your red blood cells as well. And then I'm also looking at the complex life cycle of the microbe. It has 2 hosts. So there's the mosquito host and then also the human host. And then just the fact that it's able to reproduce both sexually and asexually, depending on which of the hosts that the microbe is at any point in time, and how it's able to evade the mechanisms within the blood and can go directly into the blood cells and then cause diseases. There are some strains that can also, they don't go directly into the red blood cells, but they stay within the liver and then have the possibility of causing, a recurring disease over a period of time. So they might not infect you immediately, but they will still be there within your liver system and then cause the diseases. And then we had looked at, you said, look at how I can link sickle cell carriers with malaria disease and how they are not prone to malaria disease because of the defective red blood cell, the sickle cell red blood cell that they have, that makes it difficult for the malaria parasite to receive any oxygen and attach and then go on to cause the diseases. So that was basically the introduction on the first slide.
Oh,
And
that's way too long of an introduction.
okay, I can make it shorter. I kind of like just went over everything.
Okay, so you just have to work on your word economy. You don't want to be like explaining things live. You want to be knowing what you're going to say. You should time yourself. You shouldn't be more than 10 minutes on this. So you have to be really efficient with the words. So if I were to, go over this, say malaria, I want to talk about malaria and associated diseases of, I wanted to talk about how these microbes, Plasmodium falciparum and Plasmodium vivax, are related to the cause of malaria. If you didn't know, symptoms include high fevers, acute kidney failure, hemolysis, edema, in pregnant women, and death. So it's pretty bad. I'm sure you've heard of malaria. Let's talk about the life cycle. It has a complex life cycle, or I would just take this out, this complex life, because you're about to show them.
Okay.
So I wouldn't really worry about it. Let's talk about the microbe that's responsible for inflicting malaria in people. And so this is a unicellular protozoan. It is amorphous. It doesn't have a set structure, but it has a continuous change during its life cycle on its. And you're talking about, when you say unicellular protozoan with no fixed structure but undergoes continuous change during its life cycle, you're not meaning, what you're meaning is the surface structures.
Right.
So that's what you have to say. So I would change that, say, no fixed surface structure.
Okay.
Or this is also word economy, unicellular protozoan that has a continuously changing surface structure during its life. That's what I would put. Scisons and no one knows what those terms are.
So if I don't need to explain those, or I want to assume that since I'm talking to my class, they may have already had knowledge of the cycle that involves the schizons and the merocytes.
Nope.
Will that be relevant, or it's something that
you have to focus on what you want to talk about and then talk about that?
Okay.
You're not gonna be able to talk about everything. So you have to pick it, what you're going to talk about, and then see if it makes sense for a story you're trying to tell. These are just different cell states, right?
Right, within the life cycle of the malaria.
So I don't know. You'll have to. I don't know. I can see you telling the story without having that. But if you're going to do it, I would have to. You would probably want another slide that shows them cycling between these things, because it won't make any sense written here.
Okay, I think it's on the next slide, so I can take out that one, which has and
So this slide's complicated. If you're going to use something like this on the lower left-hand corner, so see in the lower left-hand corner where I'm at?
Let's hit the lower left.
Why don't you share your screen, share this PowerPoint, and then I can guide you on what I'm trying to show you.
Actually logging in from my phone, so let's see if I can switch it around. Okay, so this is my screen.
I don't have it yet. There we go. Oh, no, 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 no. Not like that. Let me see. You're sharing your screen. That doesn't make sense though. Like, why is
I'm using my phone. For some reason, my laptop doesn't give me audio, so I have to like either, yeah.
Oh, well, I don't know how you're gonna do this. So I guess I'll have to talk you through it. Well then never mind.
Okay.
Yeah, never mind. I'll just go back on mine. So what I would do is I would add text boxes and slowly go through these stages. If you put something like this up, it's mind numbing. You're gonna have to, I will, so what I was gonna do, I wanted you to do this, but I will have a laser pointer here and then I can follow you through the stages if you want.
Okay, I can.
This is a very busy slide and you're going to have to think about how to say this so people aren't crazy with, with the information here. So you have to say, here number one, mosquito has bitten you. Now the microbe has accessed the bloodstream. Through the bloodstream goes to the liver cell, starts to proliferate in the liver cell, and now this cell state called a scizont is in large concentrations. Once we have large concentration of the scizont inside of a liver cell, the liver cell ruptures, and then we have this microbe exposed to the blood again, and then it goes through another stage. And this is inside of a red blood cell, correct?
Yes.
So that's not, you have to make sure that you mention that to them. So here we have a red blood cell, and we have a scizont that has invaded a red blood cell, and now it's going to change again, and it's going to change in a trophozoite. Okay, and this is also known as a ring stage, if you want to tell them that. And then in here is when you start to get the further stages of development that will lead to the ruptured red blood cells and more schizonts, just like we had before. But then you also have gamete production that came from these red blood cells. And now with these gametes, the cell cycle can lead to reproduction. And so then we have, these gametes getting together, we have sexual reproduction, and then the cycle starts over again.
Okay.
It has to go through this. So you have to kind of, well, then this goes into the mosquito, right? So the
Yes.
mosquito then bites in the gametes, and I guess the reproduction happens before the mosquito.
The within the mosquito, the
So both
gametes
gametes go into the mosquito,
go into the mosquito,
and
then sexually reproduces there, and then that's what the mosquito uses to infect the human host.
Okay, so during a mosquito bite's taking in gametes and delivering sporozoites.
Okay. So I'll just rephrase make it more, reduce the words to just capture the actual pattern.
Yeah, and see if you have other slides, or say if you want to, you can break this up and you can add text boxes that are revealed every time you click it. I don't know if that's complicated for you, but anyway, so then when you have the gametes, you're like in that first mosquito bite. Now these gametes get to go in the mosquito during that event, and then these will proliferate inside of the mosquito. This is a sporogenic cycle. I won't go through the different stages, but effectively what you have is the development of these sporozoites that are in the mosquito, and the mosquito can then now deliver them into your blood during that time.
Okay.
A simple way to go through it. Don't get caught up in some of this terminology. Rally around things that they know. They know what a liver cell is. They can understand a microbe, but more, the more jargon like this macrogymnocyte, all that stuff. It's not necessary. Like, don't worry about that. I would keep this simple. Yeah, remove some of these terms. This whole
Okay.
thing, all we need to know is that the reproduction happened inside of the mosquito, and then when they're released, and now they're in the blood in the mosquito, and you're going to have an injection of sporozoites when you get a mosquito bite.
Okay.
Make it simple. Human life has 2 stages: human liver and human blood. I like that. Human cycle has its sexual reproduction. Life cycle is sexually reproduced. Why are you saying that? Oh, because there's no sexual reproduction. Okay, well, whatever. These 2 stages of sexual reproduction, right? Good. Oh, I like that. I like the way that you have simplified it on the right. If your description can be that simple,
So maybe what I'll do is I can take out the actual and then just do like diagrams to represent my bullet points
yeah.
instead of the whole CDC diagram that has.
Yeah, I think that what I like about this is that it goes from liver to erythrocyte. This could be like a simple 2-3 point arrow, though.
Okay.
Like, you can make this slide. It would be a lot easier.
Yeah.
You can, instead of showing all these things, you could just say immature, um, um, uh, plasmodium develops into whatever. Just make it simple. Don't even talk about the stages.
Okay.
Because then you would only have to say the microbe enters the liver cell, proliferates, ruptures out of the liver cell, re-enters the blood, enters a red blood cell, proliferates, releases gametes, and more intact microbes. Intact microbes can recycle. Gametes were taken in by the same mosquito. Sexual reproduction inside the mosquito leads to mature um, organisms that are available during a mosquito bite, something like that.
Okay.
So just make it simple. We can talk about this tomorrow too,
Okay.
if you want to talk about it again.
Okay. I can.
So standard treatment plan of malarial disease. This is too busy.
I can. Yeah. If it's just 10 minutes, I can take out some of the.
This whole thing would be
Just talk over it.
take out. Just
Okay.
take all that out. Yes, then just go straight to sickle cell anemia.
Okay.
Sense, yeah. Good. And that should be the end of it, right?
Yes.
Okay, I see. Okay, good. Work on your word economy and we'll go over this. Let's see. That's not, you don't need all this.
I can take out the one with the statistics. And then,
Yeah.
okay.
Good. Very simple. This is about you learning how to present. You're going to be presenting this to the students. Let's, I guess we could do this Thursday. Comes at a perfect time too. I'm about to cover diseases. Let me look at my notes.
Yay.
So going over ruminants. Let's do Tuesday.
Tuesday next week for.
Yeah.
So can I share what I'm gonna rework it and then I can.
Yeah. Yeah, we worked it. And then yeah, next Tuesday or Thursday. It's more based on where I'm going to be. I'm going to be covering Thursday. I should be through a lot of processes. So Tuesday probably sounds like a good time. I think Tuesday is going to work out because then I'm going to start going over some pathogens. That would be a great time for you to present. I think it'll
Okay,
work.
awesome. Awesome. So when can I share this with you? Maybe like in 15 minutes once I'm able to revise the whole presentation.
Oh, take your time. Yeah, we could do it tomorrow. I mean, like work on it today, and then we can work on it tomorrow, and then you can present it like you're going to present it to me, and then we'll see how it goes.
Okay. So do I need to schedule a WebEx session with you or ahead of tomorrow or?
We could, if you, yeah, let's do that. people always respond well to timelines. Let's see. Tomorrow is Wednesday. Is that right?
Or if you wanted to go over it on after Thursday's lecture, that's still fine. I can.
You can do that. That's fine.
Okay. All right. It gives me more time then.
Yeah, just be patient as people, stragglers are, talking and stuff like that. But once we're done with all the whatever they have to say, then we'll just go over what you've put together.
Okay, I will.
Okay.
All right. Thank you, Dr. Cruz.
Of course. Take care. Good job.
Thank you. All right.